Sir holds the key positions including Research Scientist for ICMR, National Director for Research at World Youth Heart Foundation, and Principal Investigator for Manipal Heart Failure Registry. Additionally, Sir is the Vice President of Medical AI Wing Indica AI Mumbai and has received prestigious awards such as the Young Achievers Award 2019 and Young Scientist Award 2020 in cardiology research. Moreover, Sir is a member of prominent organizations like the American College of Cardiology, European Society of Cardiology, and ISPO. Dr. Ajit Singh is the investigator and site coordinator for many international clinical trials. He has received several international research and travel grants from esteemed organizations, including the European Society of Cardiology, the Indian Council for Medical Research, DST, CERN, and American College of Cardiology. He has traveled to more than 10 countries regarding his research presentations and invited lectures in recent years. He visited the Madrid, Barcelona, Spain in 2022 to present his research at European Society of Cardiology. In 2023, he attended the American College of Cardiology Asia Conference and researched yoga in heart failure. Was cited by a top research newspapers. Sir has just returned from Lisbon, Portugal after presenting 3 papers on ESC heart failure 2024. Currently, Sir holds a membership in American College of Cardiology, European Society of Cardiology, and International Society for Pharmacoeconomics and Outcome Research. Dr. Ajit Singh is here to enlighten on the topic of precision medicines, appreciate for diabetic and cardiovascular diseases. With this brief introduction, I take the opportunity to welcome Dr. Ajit Singh to present his talk. Welcome, you, sir.
Hi everyone, good morning again. Okay. See, I have a disclaimer for today's presentation. So before I start, I would like to thank Sir for the wonderful introduction, sir. And your mentors as well, Srinivas from MCOPS, he was in MCOPS and one of our mentor when I was visiting my family that time he was there. So, privileged. Please chairing my session today. Thank you, sir. And thank you, everyone. So the disclaimer is that today's topic, it's a little specific if I say. Everyone is not aware of such innovative things. I'm running TeamMed and I meet thousands of the family students on a monthly basis in different campuses. They're not about the new things about such technology, if I say, and such innovative ideas of patient safety. We are still in the traditional part of patient safety and traditional part of treatments. But don't worry, I made it simple. The topic was given to me was diabetic cardiovascular complications, but I have only 3 slides on that, and whatever I will be talking about the precision medicine, so you can understand it better. Okay, so let's start. Okay, so this session is having the multiple, I have taken information from multiple sources and given the credits. I do not have any conflict of interest to declare. So today we are going to discuss from the quick questions. We are going to start with the quick questions to the audience. So you are ready, guys, for the questions? And then the background, then we'll come to the main topic, then a case study. a wonderful case study of precision medicine. You will like it, you will love it. And then I have some bonus slides which are especially for the pharmacy friends. So let's start. Questions to the audience. First question. Raise your hands, guys. How many of my audience today know about the precision medicine? And if someone can give the answer, what is precision medicine? Guys, come on. Don't disappoint me. Yes? Anyone? I mean, it's open for all. Anyone who know about precision medicine? Also, it's going to be tough for me to make you all understand the best part. Okay, no worries, I'll get it. But I think the next two questions are waste. How many of you practice the and then? Okay, this can be a good question again. How many of you have seen or heard of precision medicine practice in India? Raise your hand. How many of you have just heard of? You don't know about the precision medicine, but you have heard somewhere that okay, precision medicine is something. Yes, guys. Sir, Ramdas sir. Okay, one was there. Okay. Okay, so let me nourish your knowledge a little bit. In India, we are very rich with such tools and techniques and new innovative ideas for patient safety. You might not be knowing, but the Government of India is running multiple programs for precision medicine. I'm not talking about the individual colleges, but the Government of India has taken those initiatives and now they have started precision medicine for the diagnosis of tuberculosis at the individual basis in the rural and urban setups. We have same thing for the rheumatology. We have the same thing for the infections. We have the same thing for the heart failure cardiomyopathies in India. So it's not very new, but I believe you all have to update the knowledge, right? So that's a bit. Okay, a little background and the history. How many of you have heard of one size fits all? One size fits all. So this is the current pharmacotherapy. This quote was published 20 years back in the Wall Street Journal America. One size fits all. So the current pharmacotherapy was a one size fits all approach in which we developed the drug and we believe at least 50 to 75% of the patient will be cured. 50 to 70% of the patient will be cured. But what about the other 50 and 30%? We have no idea. So to tackle that problem, we go to the precision medicine, or we call it individualized medicine also, or personalized medicine. I mean, there's a little difference in personalized medicine and precision medicine. That is very thin line. I'll tell you that also. Okay, so if we go to the little history, I mean, precision medicine is not very new. Precision medicine is not very new. You will understand that. See, if we think of a sense, personalized medicine is not entirely new. The identification of the blood group system in 1901 by the Carl Landsteiner at the University of Vienna may be one of the first instances of recognizing a difference in each patient's biology. So what is precision medicine? Still, anyone? Based on these comments, anyone can tell me what is precision medicine? Okay, let me tell you a little more. Not now. Let's get back to the 1543, 15th century. The Hippocrates said the four humors, blood, fat, yellow white and black white, will determine the treatment of each individual. So, precision medicine, we have just a new name, precision medicine. Otherwise, individualization of the treatment. Mark this word, individualization of the treatment has begun long back, and that is the precision medicine. Let's check. If you look at this, what we observe about a patient. If I want to, precise a medicine, as we were talking about, 50 to 70% they fit in one drug. But what about the 30%? We have to know about them completely, entirely, 100%. And that 100% comes in their behavioral, social, environmental, genomics, health records, metabolomics, and genetics, and their medications. If we know about all these, we can transform or we can customize the medication treatment part for the individual. We can make the treatment pathways for the individuals, and that is precision medicine. Treating the individual based on his or her symptoms and based on his or her social, behavioral, environmental, health records, based on all the factors. I'll tell you everything about that in the next slides. So, the technical definition, precision medicine, generally considered as analog to personalized medicine or individualized medicine, is an innovative approach that uses the information about an individual's genomics, environmental, and lifestyle to guide the decisions related to their medical management. I'm not expecting you to sleep because I woke up till 4 a.m. to prepare this, okay? Okay guys, so use of precision medicine. Everywhere you can come up with a new innovative diagnosis. You can come up with the preventions to the diseases. You can come up with the monitoring. Everywhere you can use the precision medicine. You can use in the drug targets and targeted therapies, treatments. You can use in the prognosis of the drugs. I'll give the examples of that. So, how precision medicine works actually? See, I was telling about 50-70%. Usually what people do is, they recruit out the entire population. They give the drugs, they look for the p-values. How much got the positive outcome, how much got the negative outcome. Isn't it? And the clinical trial is done. We just differentiate how many are getting positive outcomes of the drug, how many are getting negative outcomes of the drug. That's all. But we don't think of why they are getting negative outcomes. We don't think of why they are getting positive outcomes. We go for the risk factors, we assess multiple things, but we don't assess entire part, right? So, first what we do is, we recruit the patients into any clinical trial, because how we come up with the research, right? That's how we do if we are starting the precision medicine. We cannot just start with one patient. We have to take a gathering, we have to take a population. And in that, we give a trial. We find out the positive part, positive outcomes. How many are responding to the drug? Or how many are responding to the intervention? How many are responding to the therapy? We get only the people who are responding well. Then we find out why they are responding well. What are the metabolic pathways, what are the treatment pathways, what are the receptors, what are the things involved, So those all things we notice. And based on that, we come up with the new drug targets, only for such populations. And then we analyze the negative part, why they are not responding, and we just don't involve them into those drug therapies. So definitely it's a long way to come up with multiple targeted drugs. But we wish that with the precision medicine, everyone will have the individualized therapy one day. Right now we are sitting in all paracetamol and the other drugs. So recruitment, we collect the data, we curate, we analyze, we report, and then we get our, as I was telling, discovery science, we know entirely about the population, and then we go to the positive part. Let's know some more. See, look at this. This is the entire public health chart for the precision medicine. See, this is not a rocket science, you all will understand. It's very simple. We talk about, when we are talking about precision medicine, we have to cover all genomics, physiology of the patient, genetics, transcriptomics, the genetic transcriptions of the patient, and what are the metabolomics, proteomics, epigenomics, microbiomics. We study the microbiomes of the body, we study the genes, we study the epigenetics, we study the proteins, we study the the entire metabolic pathway of the patient, and based on that we come to the new drug or new targeted therapy. Okay, now look at this wonderful thing. Here there are two things: population menu, which is what? Population level health data, better risk stratification. If we have many people, we can stratify more, like how different genders are responding, how different age groups are responding, how different diseases are responding. Maybe if I talk about diabetes only, diabetes has neuropathy, diabetes has retinopathy, diabetes has kidney problems, diabetes has cardiovascular problems, lot of things, right? So how the precision medicine of diabetes, if we target the diabetes only, how it is working in the different groups, so that we can come up with the group studies, we can come up with the population studies. But the best part to go to the population is one. If you have one person, if you know the one person completely, entirely, you can go to the entire population. With this help, let me tell you the story now. See, this is a case study. Biological intersections, insights from N of 1, unlocking the knowledge of N of many, from one family to the entire world. From one family to the entire world, how they have prescribed. Collaborative international knowledge sharing helped discover the pathogenic gain of function variants in mammalian target of rapamycin gene, MTOR gene, we call them motor gene. Okay, MTOR genes in multiple affected children presenting with the birth defect from the same aboriginal Australian family. So this is the story of an Australian family which has the dysfunctional MTOR genes, and almost all the kids, from the birth they had multiple anomalies, cardiac anomalies, cardiac defects, or limb defects, paralysis, and multiple defects were there in the family. So what they identified is the MTOR gene is responsible for that. MTOR gene is responsible for that. So this entire thing is known as MIND syndrome. Macrophagy, intellectual disability, neurodevelopmental disorder, which was small thorax syndrome. Due to small thorax syndrome. This is an example I am giving you, case study, how one Australian family, study of the entire family, is working with the affected children having characteristics, facial phenotype, and three-dimensional facial analysis. So what they did is, I'll come to the conclusion. Okay, I'll come to the conclusion. See, this is about the MTOR components in that sarcoma genes. These are which are now helping in developing the biological pathways for birth defects, rare diseases, and cancers. But how did they have come to this conclusion? Because they studied the entire family of with the motor gene, and then they have come to this pathway, and pathway, not only the pathway of treatment, what we are discussing here, but the diagnosis. How? Based on the family, they came up with the tool where they recognize the facial phenotype and three-dimensional facial analysis of the patient, and based on that they can say that this patient has the MTOR gene defect or MIND syndrome. So the one family, knowing the entire family, is now helping the entire world with MIND syndrome. That is precision medicine. So see, not only about the drugs, about the receptors, about the genes, about the facial expressions, about the facial analysis, everything is considered here, and they are now helping the entire world in that. So there are thousands of such case studies which can help you in that, like the precision medicine and how it can help the individual people with individual gene defects or individual problems. Okay? So this is, if we come to the diabetes, this was the topic. I have more interesting things, right? So there are three things, not three things, there are 5 things to remember, guys. There are 5 things to remember. Okay, what 5 things importantly? First is precision diagnosis, precision prevention, precision therapeutics, precision monitoring and treatment. Okay, so as I was telling in the second slide, where we can use in diagnosis, we can use in prevention, we can use in treatment, we can use in prognosis. The precision medicine can help in all every stage of the management, right? So in the diabetes, if we look at the prevention first, we have to look about the prevention, how we can stop the diabetes. So determine likely responses to the health interventions and risk factors, optimize the interventions and minimize the risk factors, exposures to the given individual. If we know the individual, what are the risk factors, what are the allergens, how he is getting the hypoglycemia, how he is getting the hyperglycemia, when he is getting the hyperglycemia, how his sugar levels are shooting up. Every factor, if we know, we can prevent that person to get the hyperglycemia or the diabetes, right? Then precision therapeutics, tailored therapy using the information about a person's unique biology, physiology, genetics and all, environmental to prevent or to treat the disease. Okay, and then precision monitoring. Monitoring is very important to understand the biological markers, behaviors, diet, sleep, psychological stress, precision monitoring. So these are the factors what you have to monitor. Okay, let me explain a little more here. This is the prognosis. So in that you can monitor all of these in diabetes, like all the diabetes related complications also you can easily understand. But will not stick here, we'll go to understand here. Look at this. The best example of the breast disease. How it works. We assess everything like the body mass index, age, gender, EGFR, simple clinical information. Other than that, physiological biomarkers, insulin resistance, C-peptide, and genetics, epigenetics, pharmacogenomics. So just a quick question to my audience. What is C-peptide and how it works in the diabetes diagnosis? Yes guys? What is C-peptide and how it helps in diabetes diagnosis? Anyone? So C-peptide is the biomarker, the plasma biomarker, which helps the clinician or the people, the doctors, to differentiate between the diabetes 2 and the diabetes 1. Okay, that's how we can differentiate between type 1 diabetes and type 2 diabetes. Okay, so based on all the things, we go for the algorithms, which is called as probability calculator, and then we look for the HbA1c response to the drugs. We give multiple drugs, and then we find out better response to drug A, better response to drug B, and some mixed responses to drug A and B. Right, based on the algorithm. We are not fitting everyone here in one set. We are not fitting everyone with one drug. Maybe someone is better with sulfonylureas, someone is better with acarbose, someone is better with DPP-4 inhibitors, someone is better with metformin. So we are identifying which drug will work with which patient exactly. We are not giving just metformin to everyone. That is the best drug, but then again is one size fits all. Right? As everybody fits all with all the BP medicines, all the BP patients. But that's not how the drugs work exactly. Okay? More precise. Look at this. More precise. The drug studies in the diabetes. See? All the features, treatments, family history, everything we have noticed, everything we have analyzed. And now this is a specialized, the MODI probability calculator. What is MODI, guys? Not going to doubt this, right? Just you can hear. Maturity onset diabetes of the young. This is a type of diabetes. Kind of type 2 diabetes, but why it is different from the type 2 diabetes is type 2 diabetes, there are two types. One is for young, one is for adults. So adults phenotype, adults phenotype does not match the diabetes phenotype in adolescent and young patients. That's why we gave them the different name called MODI. This is a type 2 diabetes only, but in young. That's why we call it MODI. Remember, this is the type 2 diabetes medicine, diabetes, but this is in the young patients because the phenotyping is different. Okay, that's why we call it MODI. So here MODI differentiation, look at this. We did all weight, birth weight, neonatal complications, glycosuria, other things, and then now we did I mean, C peptide more than 200 mmol per liter is saying that it's body. If it is lesser than that, it is type 1 diabetes. Okay? So now body genetic testing. Now look at this, how genetic testing can individualize the therapy. And look at the local scenario, current scenario what you are facing. You are going to doctors. Did you see any doctor doing genetic testing in India for diabetes? Did you ever see any one of you? No, right? If they do, they can know which drug fits where. Otherwise, they will just give metformin. If it's not responding to metformin, they will add sulfonylurease. If it's not responding to sulfonylurease, they will add DPP4. If not responding to that, they will add 10 or something else. If not, final insulin therapy. But they don't look at the phenotyping, they don't look at the genetics, they don't practice precision medicine. Look at this, simple. If the person has hepatocyte nuclear factor 1 alpha gene, that will work with low dose sulfonylurease. The best drug is low dose sulfonylurease. If the person has HLF4A for the MODY, again low dose sulfonylurease. But if the patient has GCK MODY, that is glucokinase MODY genes, that will work like there is no treatment required. It will correct by itself with the lifestyle modification. You don't need the treatment at all. That is the importance of precision medicine. That's how it works. It is costly. There are challenges. Not only one challenge, thousands of the challenges right now in practicing the precision medicine, I can say. But we have to start. We have to start with the research. And I'll tell you one more thing here. If it comes to the cost of genetic testing for one person was around 1,50,000 rupees 10 years back. But now, since it has been normalized, now the entire genetic testing for one person can be done in 35 to 50,000 somewhere. It's like it has reduced by 67%, almost 60-70%. So we just have to make it normalize. We just have to make the precision medicine normalize. Look at this. If it is HLF1B, gene, then it needs insulin. It will not be cured by sulfonylurease or metformin. But how the doctors will know about these genes without testing? They will test their drugs. They will not test the genes. They will test the drugs. Like first metformin, then sulfonylurease, then carbose inhibitors, then all the drugs. Right? We have to normalize in India. For sure it will work. Right? Now, look at this. The same thing. What are the factors we need to check? Or what are the things we can go for in different things like in monitoring, diagnosis, prevention, treatment. So, like we have to go for continuous glucose monitoring. We need antibodies, autoantibodies analysis. We need genetic score. We need C peptide levels. We need immune interventions. We need stem cell therapies. We need inflammation reductions. We need oral insulin and peptides analysis as well. We need multiple things to analyze. It is not a very simple thing. We need a comprehensive analysis for that. But it works. Until we are getting the positive response, it works. Okay? And then a lot of things are there. Now, look at this. The title, Classical versus Precision Medicine. Like I was telling how it works, how we are working. It's very simple in the classical medicine. Stage 1, one drug for the two antibodies. Stage 2, add one more drug. Stage 3, in the symptomatic disease, add one more drug. Stage 4, maybe insulin or something else. But in the precision medicine, if it is not working, then go for the prediction algorithm for the drug. How it will work, to how extent it will work. Prediction of autoimmunity, because the pancreas, beta cells, the beta cells, pancreas at least, yes. Right? So we have to check the autoimmunity of the beta cells. It's not just limited to the medicines, like medicine is not working, add one more. No, this is not the testing on the humans. This is like, if I say genuinely, what I have observed is like we are the rats right now for the, all the medicines. We we are under the clinical trials, knowingly or unknowingly. If we are getting fever, we are tested with 5 drugs. If we are getting diabetes, we are tested one by one for all the medicines. But we are not getting the right treatment. We are not getting the right diagnosis. Right? That's why we need precise medicine. I'm talking very, maybe 10 years ahead of now, but it is possible. It is possible, and it is getting possible now. Okay? And we have initiation of files, autoimmunity, prediction algorithms, interventions, monitoring, predictions of type 1 diabetes, type 2 diabetes, entire thing, right? And if we have to talk about cardiology, again, same thing, standard care of framework, a treatment that fits all, aspirin, right? All the heart attack patients will get aspirin, clopidogrel, ticagrelor, and atorvastatin with pantoprazole, isn't it? That is the standard protocol, if I'm not wrong, right? This is the standard care of framework for all myocardial wall thickening, myocardial infarction, cardiac pulmonary embolism. But if we look at the precision medicine, we have to study genomics, microbiomics, metabolomics, proteomics, and then we go for standard dose, or if we need higher dose, or if we need any altered or alternative drugs. We have to find out, right? So, and we have to also find out if we need the standard response, if the drug is giving no response, or if the drug is getting adverse response. That will come after all the things, and we will come to know why getting adverse events, why getting no response, or why getting positive response. It's not like if you are getting positive response, just give them the drug and leave it. No, just observe them, why they are getting, what are the factors linked to them, so we can individualize the same population further. Right? Now, look at this. Complicated? A little bit? Yes, guys. Yes, guys. Okay for you? You understand that by yourself, or shall I explain? Okay. So, AI, artificial intelligence and machine learning, is one of the integrative part of precision medicine. We cannot analyze the comprehensive data without the help of computers, without the help of artificial intelligence and machine learning. And that's getting possible now because we have artificial intelligence with us. Right? We have artificial intelligence with us. I talk a lot about artificial intelligence because I'm also running one of the artificial intelligence company as a vice president. Guys, just tell me. I always ask this everywhere, but it's a very important question to ask. Can a doctor, many of you, all of you at least have seen the ECG machine or echo machine, MRI machine, CT scans? Can anyone tell me a doctor all alone can develop the MRI machine? Can make the MRI machine? Can manufacture the MRI machine? Who they need? Loud. Engineers. You are saying engineers? No response? Yes? Okay. Can an engineer make the MRI machine all alone without the help of doctor? No? Okay. So what is this called as? What is this term called as? This term is called as medico-technical bridging or techno-medical bridging, where we bridge the technical part and the medical part, where the doctors help engineers and engineers help doctors in getting the applications or the solutions for the medical problems. Now let me tell you, how many of you know that IIT Kanpur has come up with a 1,000 beds hospital in their campus? Do you know that? IIT Kanpur, one of the best in IITs, now have started the hospital, their own hospital in the campus. Do you think, guys, they have a lot of patients inside? Or a lot of faculties and the students are getting sick inside the campus? Why they have opened a 1,000-bedded hospital inside the campus? They have started with 300 hospitals, 300 beds last year only. They have started a hospital with 300 beds and they are expanding it to 1000 beds hospital. Why they need it? Anyone? Why they need it? Another shocking news for you, maybe. IIT Guwahati has come up with their own MBBS curriculum. They are starting the MBBS in IIT Guwahati, and IIT Bombay also is starting MBBS. Technical MBBS, technical plus medical. I mean, MBBS will be the same, but with the integrative approaches of technology. Why they need to start the MBBS? When we have, the thousands of MBBS students coming out every year. Why they need their own MBBS people? Any answer? It's a very important part, guys. See, we'll not waste the time. The thing here is, the term I told this medico-technical or techno-medical bridging, they need it because they want the solutions in-house. They want all the clinical trials to be done in the campus. All the technological solutions they want to try in the hospital, which is available in their campus, and the patients are available for the participation. They don't want to collaborate. They don't want to go anywhere. That's why they have come up with their own hospital. So they can come up with the fastering, speed of the clinical trials and the AI-driven solutions, machine learning-driven solutions, technology-driven solutions of healthcare. That is important. Why IITs are coming up with their own MBBS syllabus and own MBBS batches? Because they felt that MBBS syllabus right now is not appropriate to fit for those solutions, AI, machine learning and all. Now they have integrated all the machine learning and AI in the MBBS solutions. That's why they need it. It is very important, and that's how we go to the precision medicine, because the technical part is important. AI is important, machine learning is important. Look at this, why? See, if you get these data, patient-specific data or public-specific data, like biomarkers, drug, diseases, variants, genes, lifestyle, phenotyping, metabolomics, genomics, proteomics, healthcare data. If you have thousands of variables to analyze.
Last 5 minutes.
I need just 5 minutes. So, if you have all of these, you cannot do it with the help of normal logistic regression or SPSS or the other data analytic tools, not at all. You need very high intensity algorithms to analyze such data. Okay, so these are the solutions what we look through the AI, like support vector machine, deep learning, logistic regressions, decision tree, random forest, linear regression, name layer, genetic algorithms, hidden Markov model. These are all the terms we use in analysis of data. And when we are analyzing the data, what we are getting at the end? We are getting all these clusters, decision support system, multifactoriate, examination, which helps us in diagnosis, prevention, prognosis, treatment and therapeutics at the end. So the importance of AI, ML in precision medicine is very important, and that's why I tell all my students, to my interns, we provide the specific AI and ML Python learnings. All families who are our intern, we give them the specific course on AI and ML and Python. So they can be, think of out of the box to learn further. That's important. Learn out of the box if you want to grow further in the field. Okay. See, how to plan the research? Simple as I told you. Just do not go with the fixed clinical trials, fixed trial designs, like we will take target population, we will take, control and cases, analyze for in the same population, same sample size for all type of clinical outcomes. No. Sample size adaptation, some people think that if we increase the sample size, we will get the good results. Definitely, you will get the good p-value. You will get the poor outcomes, you will get the positive outcomes. But no individualized results at all. You may get the p-value. You may get the good publication out of that in 10 impact factor, 15 impact factor. But that is just a clinical trial, right? But I'm definitely, again I'm telling you, I'm talking 10 years ahead from now, because right now we are doing the traditional research. But we need to upgrade ourselves. We need to upgrade ourselves. We have, Government of India has taken multiple initiatives in the precision medicine. So I always encourage our students, and I invite our students to all of you to go through the government websites where they are helping the people. They are giving a lot of grants. All the faculty members can try for it in the precision medicine, and in the trials using the AI and ML and lot of technical approaches. Right? So learning from today's lesson, the precision medicine can help in identifying the novel drug targets, identifying the biomarkers, support the design of better clinical trials, and developing the diagnostic tools and tests. Okay? Now let's come to the challenges. As I was telling, a lot of challenges. First, money. We don't have reimbursement for the patient's testing. We don't have the money for the treatments. We don't have the money for the genetic testing and all those things. To do a lot of comprehensive testing, we don't have money, right? And then healthcare provider education. Because as I asked the first question, how many of you know about the precision medicine? No one, zero. So education about the precision medicine, education about the AI, ML, education about such innovative ideas are important for the patient safety. Right? Then only we can improve to the better outcomes. And then there are a lot, right? R&D incentives, intellectual property problems. We don't have the guidelines, we don't have regulations, proper regulations for the precision medicine at all, right? Now, summary. So what we have to look for? We have to look for the right test to the right person with the right interpretations. Then only we can practice the precision medicine. Understand? Now, here is the bonus. Why we are talking about? This is the same thing that we have talked about. Now, here is the bonus. Why we are talking about the precision medicine and how you can help in that. How the pharmacist can work on the precision medicine. You confuse precision medicine can be done by engineers, can be done by doctors, but what is the pharmacist's role? Check it out, guys. Pharmacist as drug gene interaction expert. Pharmacist can identify appropriate patient and optimize the medical therapy. Pharmacist can interpret of the pharmacogenomic genomic testing results. Pharmacist can go for the pharmacogenomics consultations and educations. Pharmacist can help interprofessional collaborations in the education. Pharmacist can go for the pharmacogenomics research and guideline development. You guys can go for the global implementation and adoption of the pharmacogenomics into the practice. And if I come to the patient safety, how you can help in the precision medicine? You can identify the adults with polypharmacy and high risk of adverse outcomes. You can gather the information. You can review and recommend. You can optimize the therapies. Optimize the therapies based on the data is precision medicine. You can go for the quality evaluation and you can implement those things. Right, guys? So with that, thank you very much for the patient listening, and you guys can ask the question now. Floor is open for the questions. Thank you, everyone. Thank you.
Thank you, Dr. Ajay. The floor is open for the questions. And is there any question?
I hope I was not hard on the precision medicine. I hope you understood a bit little, guys.
If there are no questions, let's proceed. Dr. Ajit is our student, and I know him since long time, and is a very meticulous student, and he did his PhD in cardiology. He's a wonderful researcher. He has made the presentation very beautifully, and you have tried to explain almost very simplistic manner about the precision medicine and the personalized medicine. We have been talking since long time actually. Since the advent of genomics and proteins. Since then we are talking about personalized medicine, but nothing has come in practice so far. And to make it more practical now, the artificial intelligence and other technological advancement will definitely help, and definitely. I was just curious, when you were saying about the different phenotypes. Okay, and like in diabetes, you just described what are the various genes that can. So I just wanted if we can, try to relate that with our traditional system like Vata, Pitta, Kapha. Okay, and to my knowledge, even the traditional system of medicine, as you were saying about Hippocratic humors. Okay, similarly, even in Ayurveda, we have three major doshas, that is Vata and Pitta and Kapha, which is very, very highly personalized. Highly personalized. And no Ayurvedic doctor that time had one fit model. It was always a personalized this one and based on this one. So probably we can actually have a research collaboration where you can have this gene tested and test the authentic for all this.
It's a very beautiful concern, and Sir has rightly put that in Ayurveda, Ayurveda was always individualized. Always. We never had one size fits all. The people, they used to just touch your pulse and they can tell what problem exactly you have. So the problem here we are talking about is the allopathy. Allopathy, they have come up with like, okay, we need the faster solutions for the disease. If we have the infections, we don't go to the Ayurveda, right? If we have, some heart attacks, Ayurveda may not work, like immediately. It can help in the long term. So when we are talking about the fastening things, when we are talking about the allopathy especially, we always look at like, okay, if out of 150 or 60 are getting better, 40 are not getting better, it's perfectly fine. But we never are concerned about why they are getting better or why the others are not getting better. So the precision medicine, as you rightly said, sir, the proteomics and metabolomics and other things, phenotypes, we are working for so long in the basic science manner, but it's still not practiced in India. I think the first thing what I believe the major challenge is that the research is not collaborative. As you rightly put out.
Exactly, the research is not collaborative. It is always important multidisciplinary collaborative work, and only all these things will come into head. And I was not talking about the treatment part, the diagnostic part. Definitely, we can have that type of collaboration with Ayurveda and the genomics. Anyway, to conclude, it was a beautiful talk, and I would like to thank on behalf of the IPCT PS, I would like to thank you, Dr. Rajat, for sparing your time and presenting beautifully. Thank you.
Thank you. Thank you, everyone.
I would like to thank Dr. Mohammad Gulzar Ahmed and Dr. Salimullah Khan for being here for the plenary session. Thank you, sir. And I also thank Dr. Ajit Singh for their wonderful session. Now, I request our principal, Dr. A.R. Shabaraya, to present a memento and certificate to Dr. Ajit Singh. request our principal, Dr. A.R. Shavaraya, to present memento to the chairpersons. Thank you, sir.
The next session will begin after the lunch break at 1:30 p.m. I request everyone to proceed for lunch.
speaker, Dr. Grace Mary John, to take seat on the stage.
I request Mrs. Anvita, Faculty of Pharmacy, to florally welcome the dignitaries. Today, we have with us Dr. Krishnanand Kamal as a chairperson. Sir has completed his B.Pharm and M.Pharm from Manipal College of Pharmaceutical Sciences, Manipal, and PhD from Srinivas College of Pharmacy, Mangaluru. He has had industrial experience in quality assurance and R&D for 11 years. Later, in 2010, he joined as assistant professor in Srinivas College of Pharmacy, Mangaluru, and has been continuing in his role since then.
And we also have Dr. Karunakar Hegde, Professor and Head of the Department, Pharmacology, Srinivas College of Pharmacy, Mangaluru. Sir completed B.Pharm from National College of Pharmacy, Shimoga, and M.Pharm from KLE College of Pharmacy, Belgaum, and PhD from Rajiv Gandhi University of Health Sciences, Bangalore. He joined Srinivas College of Pharmacy in the year 2005 and presently working as Professor and Head of the Department, Pharmacology. He has published more than 90 research articles in the index journals. He has authored 1 book and 3 book chapters. He has received 2 times research grants from Rajiv Gandhi University Health Sciences, Bangalore.
Sir, I request you to introduce our speaker, Dr. Grace Mary John, Infectious Disease Clinical Specialist and Chief Clinical Pharmacist in Believers Church Medical College Hospital, Tiruvalla, Kerala, to the gathering and to chair the session.
Well, good afternoon. It's an immense pleasure to welcome Dr. Grace Mary John for this plenary session. Dr. Grace Mary John completed his PharmD, and she is the first and only American board certified infectious disease pharmacist in India, pioneering pharmacist-led antimicrobial stewardship programs. She is an international ambassador for Society for Healthcare Epidemiology of America and Board of Pharmacy Specialties. She has contributed significantly to the clinical pharmacy in global. She is employed at Believers Church Medical College Hospital as Assistant Director, HOD of Clinical Pharmacy. Also, she is a pivotal role in patient care, education and administration. Dr. Grace is President of the Federation of Clinical Pharmacists in India and holds key positions in the stabilization and inclusive development programs. Dr. Grace sought after the speaker. She contributes to AMS related discussions globally. She also serves as a journal reviewer for PLOS Global Public Health, bringing over 8 years of expertise in infectious disease clinical pharmacy. Dr. Grace is an Assistant Director, Infectious Disease Clinical Pharmacy Specialist and Head of the Department of Clinical Pharmacy, Washington. Ma'am is also an Assistant Professor in the Department of Pharmacology for MBBS students at Believers Church Medical College Hospitals. She has undergone antimicrobial stewardship site observation at Detroit Medical Center, Canoes Cancer Center, Detroit, Michigan, USA, and Tufts Medical Center, Boston, USA. She is a member of the International Task Force and serves as a member of the Education Committee. She has been an invited speaker for various national and international conferences, including prestigious SISCON 2023, which has held in Chennai. She is also a journal reviewer for PLS Global Public Health. So, Dr. Grace is gracing this occasion to enlighten us on the topic of practical implications of antimicrobial PKPD and dosing strategies. With this very brief introduction, I welcome you to this session. Over to you, madam. Thank you.
Thank you, sir, for the kind introduction. All right, good afternoon, everyone. So, respected chairpersons, principals, sir, esteemed faculty, and students, I just wanted to know who is there in this hall, in this hall right now. How many of you are interns, founding interns? Okay, rest of you? M-Farm. Founding? Third year? Fourth years? Fifth? There's a bunch who has not, pointed their hands. Okay, so first question, how boring do you think this topic is? The PKPD implications? Nobody thinks it's boring? No? Alright. Okay. For the PharmD students who are here, I wanted to ask you some things that I always ask my students. Why did you take PharmD? No, seriously, why did you take PharmD? Anyone? I have my reasons and I'll tell you, and you can, raise your hands if those were the reasons for you also. You will get the doctor tag. Yes? You can tell the doctors what to write. You can tell the doctors what to prescribe. So many opportunities abroad. Yes? You can specialize in certain areas after your family. Other than that, what are the reasons? These were the major reasons for me to take up this, and one other reason was that I don't have to study physics. That was one major reason why I took PharmD, because I love chemistry and I hated physics. Is there any other reason why you took PharmD? Okay? Alright. And so far in your journey, how much are you convinced that you are able to do all these things that you said, that I have mentioned, that you can tell the doctor what to write, that you are the experts, the drug experts, you can go and tell the doctor, no, this is wrong, this is right. How many have felt that way so far? Alright. Okay. I started just like any of you in this audience, okay, as a PharmD student. But one thing I wanted to do was that I wanted to be a clinical pharmacist for sure. I didn't want to go to the company side or research side. I wanted to serve with the patients, with the doctors in the medical team. So that has been my goal, and during my PharmD period, college time. And luckily I had the institution that nurtured my goals, helped me to achieve what I am today. And it took a lot of effort. It did not happen overnight. It happened over 5, 7, 8 years that I am standing here right now as a speaker to you. I had come to this college 6 years back. At that time, I was not a board certified person. I was not an infectious disease pharmacist. I was just a pharmacist, clinical pharmacist who wanted to become an ID pharmacist. Okay? So, the reason why actually they asked me for the topic, and I picked this topic because I felt that being a clinical pharmacist, your trump card when it comes to the battle with the medical doctors is your pharmacokinetics. Okay, I've learned it the hard way, so I thought I'll share some of the clinical scenarios that happens with us on a day-to-day basis, how we choose certain drugs and how pharmacokinetics, pharmacodynamics helps us in making that decision. So, like you've heard in the introduction, I am basically a person who loves antimicrobial stewardship. You know, stewardship is what is antimicrobial stewardship? The need for antimicrobial stewardship. Basically, it is the war, the antimicrobial resistance, the war we have against the superbugs. Okay? So when I started FARMACY, when I was a student, the most commonly written antibiotic, empirically written antibiotic, was ceftriaxone. Ceftriaxone is a which generation cephalosporin? Third generation cephalosporin. You know what the cost of ceftriaxone is? Approx? Okay, I'll tell you. 1 gram of ceftriaxone will cost you 50 rupees. Okay, 50 rupees. So it was one of the most commonly used empirical therapy. Now we have infections where ceftriaxone doesn't work, meropenem doesn't work, your high-end antibiotics doesn't work, and you have to go to the highest end, which is in your knowledge the highest end that you know. Way high. The highest ends for gram-negative organism. Have you heard of colistin? Polymyxin? Cephalosporin avibactam? Cephalosporin avibactam, have you heard of it? So in our hospital setting, cephalosporin avibactam is the highest. Okay, and the dose of cephalosporin avibactam is 2.5 gram QAH. 3 times you have to give the drug. And do you know the cost of that drug? Per day, easily it will cost you 20,000 rupees. Per day. Easily. So this is the difference between not having antimicrobial resistance and having antimicrobial resistance. The same infection, the same E. coli that you used to treat for 100 rupees a day is costing the patient 15,000 to 20,000. The same bug. Only thing is that they have gained some resistant genes. But you need to spend 20,000 rupees to treat that drug. So this is our war against superbugs, antimicrobial resistance. This is the reason why we need antimicrobial stewardship. So that whatever you write now, you must make sure that you know that within no time this drug also will get resistant. This drug also will become futile. So what next? You will end up with no antibiotics to treat and you will have to watch your patient die in front of you because of sepsis. Because you can't control the infection. Therefore, stewardship is one of the key roles for a clinical pharmacist, especially in ID, in infectious diseases, that we can easily do. And that's what I am doing. I am basically an infectious disease clinical pharmacy specialist doing stewardship. So, for any stewardship program, the Center for Disease Control, the center that comes up with the whole big guidelines, the COVID guidelines, the S1 guidelines, they have said that for any stewardship program to be running successfully, there has to be these 7 core elements. I just want to highlight 2 core elements in that. The second one, the accountability. The third one, the drug expertise. The reason why I am highlighting these 2 are, in accountability and drug expertise, they talk about pharmacists. Accountability can be with an ID physician or an ideal trained pharmacist. In Believers, it's a pharmacist-run stewardship program. It's not a physician-run stewardship program. Okay, in many centers, it's a physician-run stewardship program. Because as per CDC, either the pharmacist or the physician can run the program. And the third one, the drug expertise. They say that if your stewardship has to run successfully, you need an expert of drugs. Now this is a multi-million-dollar question for the clinical pharmacist. How can we become the drug expert? Right? We like to call ourselves, even doctors think just coming out of the pharmacy course, oh, you know everything about drug. The reality we don't know anything about the drug. Right? So from there, how do we build ourselves to become the drug expert? So that's why I thought this PKPD, the pharmacokinetics dynamics, is one area. If you home in on this area with your clinical practice guidelines, you will be way above your colleagues, your doctors. They will not be able to argue with you if you know your kinetics well. So certain examples I have. Again, the reason for stewardship is that, as you can see in this picture, in the 1980s and in the 1990s, there used to be a number of new antibiotics that comes to the market. Okay, right now we don't have many drugs because how much time does it take for a new drug to come into the market from drug development? How many years? How many years? Easily 15 to 20 years, 10 years minimum. It takes, no? So 10 years of investment for the company, and the moment you introduce that drug, the bacteria is very cunning that they become resistant. Okay, and within 10 years, again, your drug is out of the market because it can't be used because it is useless. So why should the pharma industry invest in antibiotics when they can invest in oncology, when they can invest in diabetics, where you have to take the drug lifelong? Okay, so the return of investment is not there for antibiotics. Therefore, again, the problem is that we don't have antibiotics and the resistance is increasing. So the one good thing that we can do is to make sure to rationalize your prescription by exploiting the pharmacokinetics and dynamics. So you know the right R's, right? The R's, how many R's are there? Right patient, right drug, right dose, duration, administration. And when it comes to stewardship, there is one more thing, the right pharmacokinetic and dynamic parameters. So that the patient that you are treating will have minimal harm, and for the future patients, the resistance is not transferred by optimizing your therapy. Now, just wanted to touch through the basics because 4th years, M.Pharm and 5th year, 6th years, you want studied kinetics, right? 4th year is your kinetics, right? What is kinetics? Oh, that four letters, ADME, what is it? Yes, absorption, distribution, metabolism, excretion. In simple term, what the body does to the drug. And what is dynamics? Which is either the pharmacological effect or the toxic effect. Right? That is clear? Yes. So, when you administer a drug, as you can see, over time, concentration, slowly the drug is absorbed, then slowly it gets distributed, alongside clearance will happen, metabolism will happen. Okay? So, this is a drug curve. Now, why are antimicrobials different from the other drugs? This is the traditional pharmacology. There is drug. What the drug does to the patient is dynamics. Patient does to the drug is kinetics. Why is it different with antimicrobials? Yes, there is a third compartment. Bugs. the PKPD has to work for the drug, not just for the patient but for the bug also. So it is not a two-way traffic, it is a triangular, it's a triad that we have to address. And how many of you know about MIC, minimum inclusion concentration? Yes? What is it? So basically, if you have, in each of these test tubes, there is the same amount of bacteria. Okay, same amount of bacteria, but each test tube will have serial dilutions of antibiotics. So in the first test tube, the least concentration, then double, then double, then double. So MIC is nothing but when there is no visible antimicrobial growth. That means the drug is able to visibly inhibit antimicrobial growth. This is MIC. The reason why I am taking these basics for you is because my talk is going to be based on these things. Okay? And obviously you know the different categories, classifications of antibiotics, right? You have your chemical classification, which is beta-lactam, cephalosporin, fluoroquinolones, all these things are there. And mechanism-wise also you have. So, no matter what you do, when it comes to antimicrobial therapy, it's very difficult to achieve your targets. So, one way of exploiting pharmacokinetics and dynamics, this is one best example that everywhere is being quoted, is the continuous infusion versus the intermittent infusion versus the prolonged infusion. I understand you don't understand the thing of what I said just now. I'll come to that. Before getting into that, you've all gone to the hospital, right? You've all seen cases, right? You've all seen antibiotics being written. Okay, let's take ceftriaxone or meropenem. Which antibiotic do you see commonly? IV. Ceftriaxone. Okay, what is the usual dose? 1 gram? Yeah, OD is there, BD is there. So how do you give that 1 gram? IV as? Infusion over? Over how many hours or how many minutes? So basically I am asking you, when you want to give an IV drug, what is the standard infusion time? Over what time do you give that infusion? Different drugs, it will be different. Like vancomycin, because of the toxicity, you have to give it over 2 hours or so. But usually, the standard infusion time, anyone? 30 minutes. Alright? So usually, 30 minutes is the standard infusion time. So here we are introducing a new concept which is purely based on pharmacokinetics and dynamics. Okay, so this one you understood, right? When you give a drug, the drug absorbs, or if it's IV, the concentration is there, then metabolism, distribution and excretion will happen. Okay? So, this is your x-axis, this is your time axis, and this is your drug concentration axis. Clear? And you have a bug with an MIC of, let's say, that dotted line is your MIC. If you give a drug below the MIC, it will not work. That's clear, no? Because it can't inhibit growth. It has to be above the MIC. 4, there are 3 classifications based on pharmacokinetics for antibiotics. I will come to that. Time dependent, concentration dependent, and AUC by MIC dependent. I'll come to the time dependent one. So the concept is very simple. So here you have, this is the MIC, okay? And how much drug is there above the MIC? Only this much, right? And how much time is it? About this much time, right? Half of the time the drug is above the MIC, after that it falls below the MIC. Is that clear? Yes. So this happens with standard infusion. When you give over 30 minutes, all of a sudden there is a peak, and soon after that the dissolution excretion takes place and it falls below the MIC. So there are a classification of drugs, basically the beta-lactam antibiotics, all your penicillins, cephalosporins, they come under time-dependent drugs. So, it means that how much time you can extend. Like in that graph I showed you, at one point it goes down, right? But is there any way in which you can extend that time above MIC? So 3 ways you can do it. Either you can increase the dose. So the peak will be high, slowly it will come, right? Second, you can shorten the duration. Instead of giving it as Q8H, maybe you can give it as Q6H. So intermittently you give, so the drug doesn't fall below the MIC. Or the third thing is, instead of giving as a 30-minute infusion, you prolong the infusion. You give it over 3 hours instead of 30 minutes. Okay, this is the third way of doing it. If you increase the dose or increase the frequency, toxicity can also happen. So the best way, easiest way is to increase the prolonged time. Now I will tell you, there are 2 ways of prolonging infusion. Either you give it continuously. Okay, that means if meropenem you want to give 1 gram Q8H, in one day you are giving total of 3 grams. So instead you give 3 grams over 24 hours. You do it, that is continuous infusion. Or there is something called as extended infusion where instead of giving for 30 minutes, you give it for 3 to 4 hours. You extend the infusion time. Now I will show you the graph. So the red one is your standard graph with 1 drug administration. Soon after, you can see here around 4 hours time, the drug falls below the MIC. Because this is the MIC line. This grey bar is the MIC line. At 4 hours, the drug falls below MIC. Now, if you give it instead as a 3-hour infusion, you don't get a peak. You don't get that dose dumping all of a sudden. Instead, slowly the dose is being entered into the body. Therefore, what happens? Slowly, the drug falls below the MIC. You get additional 2 hours. Okay, for continuous infusion, it never goes below the MIC. Okay, now you tell me which is the best way of giving the antibiotic to a patient for such antibiotics. Continuous infusion. Okay, second best, prolonged infusion. Okay, and least is your usual 30-minute infusion. This concept is clear, right? You can classify drugs based on this PKPD parameters. Drugs that work best when you increase the time above the MIC. Drugs that act best when you increase the concentration all of a sudden. And the third category where there is a mixture of both. I will come to that. So this is one example of meropenem that was done. And you can see here, like I said, this one is the standard infusion. 30 minutes, the drug falls below MIC at 4 hours. But when you give it as extended infusion, you get additional 2 hours. Okay. I will just skip through this one. So, prolonged infusion is a way because it has been proven that it helps in reducing the mortality, favorable outcomes with total daily dose, even length of stay and cost effectiveness. Because the patient is getting better much earlier than before. Okay? But there are some practical implications that if you want to give it as a continuous infusion, you need one IV line just for that. And it's practically impossible to have one IV line just for that. So that is the reason why continuous infusion is not so much advocated for, but instead you prefer giving extended infusion. Then again, stability. Meropenem, if you look at the label, meropenem once reconstituted, the stability is only for 4 hours. After 4 hours, it decreases. So you can't give meropenem over 24 hours. Okay, so that is also out of the question. So all these things matter, and there are incompatibility with other drugs. I will skip through the incompatibility you shouldn't listen to me. Even if it is so sugar-coated that the concept is very, attractive, unless it is evidence-based, you should not enter into such practices. So what is my evidence? There was the Blink 15, in 2015 there was the Blink 2 randomized control trial which said it is not so effective. In 2017, 2018, two meta-analysis said yes, when you give antibiotics, especially piptaz and meropenem, as extended infusion, it has better benefits. Even in surviving sepsis guidelines it came, but the latest trial that came in JAMA, the Journal of American Medical Association, said not that it is bad, but it doesn't have much effect. But we are awaiting the Blink 3 trial, which has the highest population of patients, 7,000 patients, head-to-head comparison for meropenem and piptaz, and we will await that trial. And most likely, I hope the trial comes up with the data saying that yes, please give it as extended infusion. Now, in the meanwhile, this is just read through this topic, this heading. This is a guideline. Okay, this is an international consensus endorsed by American College of Clinical Pharmacy, British Association of Antimicrobial Chemotherapy, Cystic Fibration Foundosis, ECMID, IDSA, SIDP, SSEM. All the societies came together to address this issue, whether to give beta-lactam antibiotics as extended infusion or not. So this is the evidence that I am telling you. All these societies came together and they suggested that yes, when you have a severely ill patient, the data suggests that for gram-negative infection, you treat the patient with extended infusions than standard infusions. So this is like a low-hanging fruit of stewardship, that you are not telling your doctor, no, this is wrong, right. You are suggesting to them something that is better, that can have better survival rates. You tell them, sir, instead of giving 30 minutes, can be administered over 3 hours. You are not conflicting anyone. But this is purely based on pharmacokinetics. This is based on the time-dependent effect of the antibiotic. But they say that for non-severely, the patients who are in ward, who are not so sick, may not be. You don't need to give it over 3 hours because the data is not so robust. Because other practical problems are also there in what, how long can you, rent this syringe pumps and other things. The logistics won't work out in for non-sick patients. For ICU critical care, yes, always give meropenem test as 3-hour 4-hour infusion. And whether loading dose, so the first dose that you give is a loading dose, right? So the loading dose, whether you should give it as 3-hour infusion or 30-minute infusion. It has to be given 30 minutes. It has to be rapid. Even if not even 30 minutes, some drugs you can even administer over 2 minutes. Okay? Loading dose is very essential so that your drug goes above the MIC. Okay? Unless your drug goes above the MIC, if you give it slowly, it won't, that effect is not prolonged. So the first dose, never ever compromise your loading dose. Never ever tell your nurse to give the first dose as 3-hour infusion. Always tell them the first dose should go within 30 minutes. Second dose onwards, subsequent doses, you have to give it over 3 hours. Alright, now this was a scenario that I used to face a lot because you have all these CKD patients coming with sepsis and all other kinds of things, and your doctor wants to give piperacillin tazobactam. Okay? Based on the CKD thing, there are these two doses. The dosing per day is the same. You either give it as 4.5 gram 2 times a day or 2.25 gram for 4 times a day. Which one would you suggest? Which one would you suggest? For piptaz, meropenem, does the peak matter? Does the peak matter? The first loading dose has to be 4.5, no doubt about it. Does the peak matter for time-dependent antibiotics? It's the x-axis that matters, the time that matters. So instead of giving 2 doses like that, it's better that you give it more frequently. So when you have such scenarios, it is your clinical pharmacy intervention to tell them that because it's a time-dependent antibiotic, instead of giving it as BD, it's better to give it more frequently so that your drug stays above your MIC. Okay? So this is concept number 1. So piperacillin, tazobactam and molnupiravir for critically ill patients, always give us 3 hours, always give the loading dose, and when it comes to piptas and molnupiravir, if you have more frequent dosing, go for the more frequent dosing than higher doses once or twice a day. Alright, now there are 2 more categories of PKPD classification, that is the concentration dependent antibiotic. So I said for time dependent antibiotics, the x-axis matters. Only this axis matters for time dependent. But when it comes to concentration dependent antibiotics, only this axis matters. It doesn't matter whether it falls down the MIC, because concentration dependent antibiotics, mostly they come under the category of protein synthesis inhibitors. If you look at antimicrobials, can you give me some examples of protein synthesis inhibitors? Aminoglycoside, tetracyclines, chloralphanicol. What are they? If you look at the time dependent, most of them are cell wall inhibitors. See, the good thing about protein synthesis inhibitor is that it goes and inhibits the 50S or 30S, right, ribosome, and it inhibits protein synthesis. So even if the drug concentration falls below MIC, for the bug, it takes time to reproduce that proteins. So that is called as a post-antibiotic effect. Even if that one dose that you give, even if it falls below MIC, that drug continues to act, because for the bugs it will take more time to become back to normal. Alright? So the best example that I can give you, as you can see here, is your aminoglycoside. So the various studies have said that, showed that aminoglycosides, earlier when I used to practice aminoglycosides, for example, amikacin, it used to be given as 5 mg Q8H. Okay, it used to be given over 3 times a day. Now, the guidelines suggest that it's a concentration dependent antibiotic. The x-axis doesn't matter, for them the concentration matters. Therefore, all together you give it as OD. So instead of 5 mg Q8H, the dosing for aminoglycosides is 15 mg per kg OD. This is for patients with normal renal function. So I am just introducing the concepts to you so that you can go and learn about it further. And then there is a mixture of other drugs like fluoroquinolone, vancomycin, where it's a mixture of both. Okay, it is the area under the curve that matters over MIC. So how much that comes here matters. Both the concentration and the time matters here. One good example that I can give you again is vancomycin. There was this landmark study that said that if the AUC by MIC ratio of vancomycin, so basically if you need to know AUC, MIC you get it from microbiology lab for your Staph aureus MRSA. Okay, my MIC is 2. Okay, the lab is giving me MIC of 2. For you to measure the AUC, you need tedium. So if you have tedium and if you measure the MIC, this is the study I am talking about, not that it is practical in every case in your hospital. But in the study what they did, they measured the AUC, area under the curve, and if the area under the curve by MIC ratio was above 400, it showed that there is better chances of clinical efficacy rather than AUC by MIC ratio of less than 400. That is the study. So one simple thing that I can ask you. Let's say the AUC is 400, okay? And you have your lab report saying that your MIC is 1. So AUC by MIC is 400 by 1, which is good enough. You can continue to treat. Okay. Next patient, AUC is again 400, but the MIC of Staph aureus for vancomycin is 2. What is your AUC by MIC? 200, which helps us predict that that patient will go into clinical failure if the same dose is being given. Instead, you can hike up the dose so that you get the AUC by MIC about 400. So that was the landmark study. So with that, I will just finish the PKPD classification of antibiotics. So I have a case for you. We have an 80-year-old patient, obese patient, 82 kilogram, height is only 152, hypertensive, diabetic, 25 years with lower urinary tract infection, creatinine of 1.4. Culture shows a gram-negative bacilli and sensitive only to amikacin or gentamicin. So, how do you calculate your creatinine clearance? CG formula? Okay, this is one formula every pharmacist should be able to say even from their sleep. Okay, 140 minus H into 140 minus H into weight divided by creatinine by 72. Alright, so like I said, for this patient, my physician, the creatinine clearance was 50 and my physician advised the dose. I told you unknown glycoside, the dose is 15 mg per kg once daily. So 15 mg per kg for this patient, it came as 1 gram. Okay. Luckily, she called me, my physician called me and asked me, Grace, could you please check the dose? Alright? And I'll tell you what happened. Problem with weight-based dosing is we have 4 body weights. Do you know that? One is total body weight. If I go and stand on a scale, what does it show? That is my total body weight. Ideal body weight. Okay, I am a bit obese. So for my height, I should be around maybe 75 kilograms. Okay, that is my ideal body weight. There is adjusted body weight. What is adjusted body weight? Let's say for a person, if 50 kilograms is their ideal body weight, but that patient is weighing 80 kilograms. Okay, 30 kilograms is extra. So do you think that 30 kilograms is fully fat? Is it fully fat? That extra kilogram? Usually, in that extra 30 kilograms, 40% is muscle loss. The rest is fat.